Hello, today I will introduce you to the cell inserts of Nest. Cell inserts are commonly used in co-culture experiments, chemotaxis experiments, cell migration experiments, cell invasion experiments, and drug transport experiments. First, let's take a look at the structure of a cell insert. Then I will introduce you to the principles and purposes of different experiments. Cell inserts are commonly used in cell experiments, such as co-culture experiments, chemotaxis experiments, cell migration experiments, cell invasion, and drug transport experiments. These permeable supports may effectively improve the culture of polar cells, since these supports allow cells to secrete on and absorb molecules from their basal and apical surfaces to metabolize in more natural ways, as well as to simulate in vivo environment to the maximum extent of culturing and some special cell lines. The cell insert unit is composed of inserts and a plate. The insert is in the shape of a small cup with a microporous membrane at its bottom, and the part above the membrane is called the upper chamber. The part below it is called the lower chamber. The difference between the membrane and the plate bottom is around 0.9 millimeters. How does such a model help? Firstly, the cells inoculated in the upper chamber grow on the microporous membrane, so they can intake nutrients from below through the pores of the microporous membrane. This way of obtaining nutrients is the same as the way the cells use to getting nutrients in vivo, which is different from 2D culture. 2D culture refers to the culture of the general TC-treated surface. Its disadvantages include that it cannot stimulate the in vivo growing environment and a lack of interactions with the extra matrix during cell production. If the cells inoculated in the upper chamber are polar ones, like epithelialytic cells or endothelialocytes, then their growth in the upper chamber will have polar differentiations, showing polarity functions. This is different from the general 2D culture. From this picture, we can easily see that the cells growing on the TC-treated surface are spreading out with very weak intracell connections. The figure below shows the growth status of the cells inside the cell insert. The cells have grown into a shape of column and show polar differentiations. The intracell connections are very close, forming interior and exterior barriers, which may help cells show polarity functions. What is cell polarity? The 3D morphology of cells, especially the ones within tissues, are often not random, or even instead, they will present axialization, namely the morphological property of the molecular structure in the cells always distributes evenly along a certain or several axial directions. Then these cells have polarity. Secondly, the cell insert divides the whole cell culture area into the upper chamber and the lower chamber, so we may inoculate one type of cells in the upper chamber and the other one in the lower chamber during the co-culture of several cell types. The growing environments of the two cells are totally the same, since they may communicate and exchange through the microporous membrane. Meanwhile, the two cells can be separated easily. Therefore, the cell insert is a good tool for the cell co-culture. Thirdly, thanks to the existence of the microporous membrane as a barrier, we are able to study the transportation, secretion, and diffusion of some substances inside the cell, including small molecules, large molecules, and even viruses. Next, we will be talking about the membrane materials of cell inserts. The membrane on the cell insert is formed by physical and chemical processes. The former determines pore density and the latter the pore size. Nest cell inserts have two membrane materials available, PC membrane and PET membrane. The PET membrane has better transparency, making it easier to observe cells. Both of the two membranes are semi-transparent to the naked eye, but the PET membrane shows better transparency when observed under a microscope. The PET membrane is naturally hydrophilic and does not require special treatment. The PC membrane surface will become hydrophilic after treatment. The protein binding capability of the PC membranes is lower, which may reduce protein loss. With respect to dissolution resistance, the PET membrane performs better. The PC membrane has poor resistance to strong bases and oxidizing substances. The PC membrane has poor resistance to strong bases and oxidizing substances. The PC membrane has poor resistance to strong bases and oxidizing substances. The PC membrane is better in terms of membrane tenacity. These are our real shots. The contrast is not very obvious on the display, but if you observe them under a microscope with the naked eye, the difference will be very significant. Next, let me introduce the characteristics of cell inserts. What's the role of the small window on the cell insert? This design is mainly to facilitate liquid adding into the lower chamber by tips or pipettes, with no need to take the insert out. The insert also has high universality and are suitable for cell culture plates from any brand. The membrane of our cell inserts have multiple pore size options. The membranes with a small pore size are designed to prevent cells from passing through, and the ones with big pore size allow cells to pass through. Normally, the earlier one is used in toxicity trials of some cells, while the latter is generally for migration trials of tumor cells. The commonly used pore size are 3 UM, 5 UM, and 8 UM. This is the market solution of cell inserts. The products from Corning, Falcon, and Millipore have relatively high market shares, and our design is close to that of Corning's. As we can see, the cell insert from Millipore has no holes on it, making it less convenient to pick up. The window for liquid adding on the Falcon product is a bit too small. This is a selection guidance of cell inserts. We recommend products with different pore sizes based on the applications and cell types. Next are the application models of cell inserts. The first are the toxicity tests. The approach is to inoculate the cells on the upper surface of the membrane in the cell insert to form a skin-like structure. Then add the drug to be tested into the insert, and add the fluorescent dye after a period of incubation to detect the cell viability. The cell viability reflects the toxic effects on the substance to be tested on the cells. Next, we'll look at the transportation model of cell inserts. The approach it adopts is as follows. Inoculate cells in the upper chamber to let them grow into a dense and fully fused cell monolayer. Then add the drug to be tested into the upper chamber, part of which will be transported by the cells into the lower chamber, and perform detection. The results will show the transportation status of the small molecule drug in the cell. The cell inserts can also be used for studies on cell chemotaxis, migration, and invasion. Cell chemotaxis refers to the response of cells in the form of movement to chemical stimulation at a certain concentration. The cell migration refers to the movement of cells from a place to another, either random or directional. Chemotaxis experiment. Cell invasion refers to the cell movement through a barrier as a response to stimuli with a concentration gradient. For chemotaxis experiment, first inoculate cells into the insert to form a dense monolayer. Then put a substance which can induce cell chemotaxis into the lower chamber. The cells pass through the membrane pores under the effects of these chemotaxis inducing substances and enter into the lower chamber. The capability of cells to show chemotaxis under effects of the chemotaxis-inducing substances may be determined by counting the cells in the lower chamber. Cell invasion experiments can also be performed. For this experiment, apply a layer of gel on the membrane in the cell insert, first to form an extracellular matrix barrier. Then inoculate tumor cells on it to form a dense monolayer. Similarly, add chemotaxis-inducing substances in the lower chamber. The tumor cells will go through the gel layer and enter the space below the membrane under the effects of these substances. We can then determine the evasion capability of the tumor cells by counting the cells in the lower chamber. Next, we will learn about another application model, co-culture. Inoculate cells in the upper and lower chambers of the cell insert, respectively, for co-culture. The common applications of this model of co-culture include lymphocyte stimulation reactions and trophoblast culture of stem cell culture. Cell inserts can also be used for the construction of blood-brain barrier. The so-called blood-brain barrier refers to a highly selective semi-permeable which separates the circulating blood in the brain from the extracellular fluid in the central nervous system, CNS. To establish a blood-brain barrier model, turn the cell insert over and inoculate one type of cells on the lower surface of the membrane. Then turn the insert back over and put it into the cell culture plate, and then inoculate the second type of cells in the insert. The two cell types form a symbiotic status. It is very critical blood-brain barrier model. Finally, the main client groups of cell inserts include tumor drug research institutes, R&D departments of biopharmaceutical companies for anti-tumor drugs, industrial clients, institutes of Merida Medica, and tissue engineering teams of stem cell companies.